The processes of transcription and translation, a short video on what goes into prokaryotic transcription and translation. By the end of this video, students should be able to define the processes of both transcription and translation, know the main sequences for transcription, know what goes on during each step of transcription, know how proteins are related to translation, know and be able to distinguish the key differences between prokaryotic and eukaryotic transcription and translation, be able to fill out a short worksheet provided, and lastly, be able to transcribe a gene that is given to you. Today, we're first going to begin our discussion on the process of transcription. Transcription is simply defined as the process of taking information contained in DNA and copying it into RNA nucleotide sequences. Before we can begin to get into the nitty-gritty details of the steps of transcription, it is important that you know and understand the main influences and sequences that are involved in this process. So the two main influences involved are the promoters and the terminators. Promoters are DNA sequences that signal RNA polymerase where to bind and begin transcription and are always located in the upstream promoter region. Terminators are DNA sequences that tell the RNA polymerase where to stop transcribing. In the image down below, the promoter is that green region and the terminator is the blue region. The promoter will always be located first and the terminator will always be located last on a gene sequence. And then in between the two is going to always be the coding sequence. This is the sequence that's going to get transcribed into RNA and then later translated into amino acids, but we'll talk about that a bit later. It is now time to discuss the main sequences involved in this process. It is important that you understand these fully so that when we go and discuss the steps of transcription, you understand their roles. The first main sequence we're going to discuss today is the open reading frame. The open reading frame is the portion of the RNA that when translated into amino acids, it does not contain a stop codon. The next important sequence is the regulatory sequences. These are the ribosome binding sites and consist of the repressors and the activators. This influences whether or not the RNA polymerase can bind and start the process. The next important sequences we're going to discuss are the untranslated regions or UTRs. The 3' UTR has no function in transcription, but is very important in the process of translation. The 5' UTR is located in the ribosomal binding sequence and contains the Shine-Delgarno sequence. The Shine-Delgarno sequence is 5 to 9 nucleotides preceding the start codon in prokaryotic messenger RNA that is recognized by the ribosome as the correct site for binding the mRNA molecule prior to the start of translation. The next important sequence we're going to discuss is the +1 gene. The +1 gene is where the transcription actually begins, and it will always be located upstream. The last important sequence we're going to discuss today is the -35 and -10 region, which contains a Tata box, which is where the sigma factor is most attracted to and will bind. It is important that you know where these sequences are located on an actual gene, so I'm going to demonstrate their locations now. There are two sequences or strands involved in this process, which are called the coding strand and the non-coding or template strand. We'll discuss later on, but only one of these strands actually gets transcribed into RNA and then translated into amino acids. The first important sequence involved is our upstream promoter region where the RNA polymerase will actually bind. The next important sequence is a sequence that we already discussed, but it's that -10 and -35 region where the Tata box is located and the sigma factor is most attracted to and will bind. Next is our +1 gene, and right down the +1 gene is our start codon. In DNA, it's called ATG. However, in RNA, it must be translated to AUG. The thymine must be traded for uracil in order for the RNA polymerase to be able to read it and begin the process. Down here is actually going to be our RNA sequence strand. And then right here is our 5' UTR region, which contains that Shine-Delgarno sequence. Another important sequence is our terminator sequence. This is going to tell the RNA polymerase to stop doing its job and that it's time to hop off the strand. And usually in the terminator sequence is our stop codons. And our last important sequence is that 3' UTR region that has no function in transcription, but is important in the process of translation. It is now time to shift our discussion into that template versus non-template strand I just mentioned before. So there are two strands associated with the process of transcription, the template and the non-template or coding strand. The template strand is the strand that will actually get transcribed to RNA. The non-template strand has no direct function during the process of transcription. The RNA sequence strand, which was that bottom strand that I drew very last, is the result of base pairing the RNA to the template strand, which mimics the coding strand. In the image down below, you can see that the coding strand is up top, this strand, and our template strand is on bottom, this strand. So this strand right here, the template strand, is the strand that's going to actually get transcribed to RNA later on. And as you can see, the RNA sequence right here is base pairing to the template strand, which will mimic the coding strand down below. Now, I mentioned earlier that the top box is where the sigma factors is most attracted to. So now I think it's important to discuss what exactly sigma factors are. Sigma factors facilitate the binding of the RNA polymerase to the promoters. There are three main types of sigma factors we're going to discuss today, and those are sigma factor 70, sigma factor 32, and sigma factor BS32. Sigma factor 70 guides the RNA polymerase for most genes. This is going to be the sigma factor we're going to continue our discussion on later today. The next important sigma factor is sigma factor 32. Sigma factor 32 is active when the cell is stressed by heat, and it is considered a heat shock response. Lastly, sigma factor BS32 is also active when the cell is stressed by heat. So just like not both strands get transcribed into RNA, not all genes are going to get transcribed or translated either. There are two different types, structural versus non-structural genes. Structural genes are the genes that get transcribed from DNA to RNA and then get translated into amino acids and go on to become a protein. Non-structural genes, however, get transcribed from DNA to RNA but do not get translated into a protein. So therefore, they never go on to step 2. They only stop at step 1 of transcription. Now that we got all of our background information done, it's time to discuss finally the steps of transcription. So transcription, as mentioned before, is the process of taking information contained in DNA and copying it into RNA nucleotide sequences. This process takes place within the nucleus of prokaryotes, not the nucleus like eukaryotes, since prokaryotes don't have one. There are 3 stages of transcription that we're going to discuss, and these are the initiation, elongation, and termination. So our first step we're going to discuss is the initiation step. During bacterial initiation, RNA polymerase binds the promoter, which creates a closed complex. When the Tata box located in that -10 region unwinds, the complex becomes open and the sigma factor, typically 70, is released. Then the core RNA polymerase slides away from the promoter and elongation can begin. During elongation, this is the process where RNA is actually made. The RNA polymerase only transcribes one strand, like mentioned earlier, which is the template strand, not the coding strand or non-template strand. The open complex is about 17 nucleotides long, or strands long, and behind it, the DNA rewinds into a double helix. RNA is then synthesized in the 5' to 3' direction using ribonucleosides and triphosphates as precursors. RNA is then synthesized at approximately 43 nucleotides per second. The last step that we're going to discuss today, and the last step in the process of transcription, is termination. During termination, the RNA-DNA hybrid is forced to separate and the newly synthesized RNA gets released, as well as RNA polymerase. E. coli has 2 different mechanisms for termination, which are the row-dependent termination and the row-independent termination. Row-dependent termination requires a protein known as Rho or R. Row-independent termination does not require R. So now I'm going to go in a little bit more detail about these 2 types of termination because it's very important that you understand the differences between these 2. The first termination we're going to discuss is the row-dependent termination. Row-dependent termination requires a protein known as R, like mentioned previously, and during this type of termination, the location where the row protein assembles, the RNA synthesizes a stem loop at the terminator. This is known as row utilization or RUT, R-U-T. The stem loop then can perform a jerking effect, which dislodges the RNA polymerase and effectively ends transcription. However, row-independent transcription does not use the row protein. It is instead facilitated by two sequences in RNA, a uracil-rich sequence located at the 3' end of RNA, and a stem loop structure just upstream of the uracil-rich sequence. This creates a uRNA and an A-DNA hydrogen bond, which is very weak. This works because of the inverted repetition where the stem loop complementary sequences are heavily attracted to each other. Like in row-independent or row-dependent termination, the stem loop performs a jerking motion and detaches RNA polymerase, which signals the end of transcription. Now that we discussed transcription fully, it is time to talk about the process of translation. Translation, the simple definition of it, is the process of synthesizing polypeptides from the RNA nucleotide sequences. Just like transcription, translation also takes place within the nucleus of prokaryotes. It is not split in different locations like in eukaryotes. Translation can start in prokaryotes before transcription is even over. This does not happen, by the way, in eukaryotes. This happens because transcription creates multiple mRNAs from a single gene, which this is known as polycentronic mRNA. Ribosomes then go and bind the mRNA while they're still being synthesized. This allows for multiple proteins to be made rapidly from each mRNA, which is an advantage of not having a nucleus. That whole process is called the coupling of transcription and translation. Now we're going to move on and discuss the steps of translation. Just like in transcription, there are 3 steps known as initiation, elongation, and termination. All of these steps require additional protein factors, and both initiation and elongation require energy, typically in the form of GTP. During termination, release factors somehow recognize the stop codons, which modify the ribosomes to activate ribozymes, which sever the polypeptide from the final tRNA. The ribosome then dissociates into subunits. So both of these processes are ongoing, and since they're ongoing, they need to be controlled. Only about 75% of genes are expressed at full times, and these are known as constitutive genes. The rest are regulated so that they are only transcribed and translated when the cell actually needs it to save energy. The cell does not want to be transcribing and translating genes that it does not need because they already have or it's not needed at that time. That would be such a waste of time, energy, and resources. This regulation can be seen in the regulation of protein synthesis, which typically halts transcription and can stop translation directly. During translation, proteins can be secreted in different ways. 3 of those ways are through a signal sequence, a seq-dependent pathway, and a type 1 secretion system. A signal sequence is an N-terminal amino acid that is bound by the signal recognition particles, or SRPs, which targets the ribosome to the plasma membrane. The seq-dependent pathway delivers proteins to the periplasm. Lastly, the type 1 secretion system secretes proteins out of the bacteria. Once the proteins are secreted, they can be modified by enzymes. So once proteins are secreted, they don't have to stay the way that they were originally secreted. They can be modified and changed. So a couple of these examples of protein modification that goes on within an E. coli or a bacterial cell is when FMET is removed from the N-terminus. FMET is N-formal methionine, which is the start codon for prokaryotes. We'll discuss this a little later, but that's what FMET stands for. Another example of protein modification is when small groups, which are phosphoryl groups, methyl groups, and adenylate groups, are added to amino acids. Another example of protein modification is when proteins are cleaved, or lastly, when proteins are refolded. Proteins can be refolded in 2 different ways. The GRO-EL and GRO-S complex can refold denatured proteins using ATP, or proteins can be refolded to fit into a center, which is known as the barrel shape. Protein folding is just basically when the protein structure is determined by amino acid sequences. So now that we've discussed in full the processes of transcription and translation, it's now time to discuss the differences between prokaryotic and eukaryotic transcription and translation. We'll first begin this discussion with the differences between gene and RNA structures. Our first gene and RNA structure we're going to discuss is ribosomal RNA. Ribosomal RNA is different in prokaryotes and eukaryotes. In both, they consist of two subunits, a large and a small subunit, but the subunits are different. In prokaryotes, the large subunit is 50S and the small subunit is 30S. Together, the rRNA for prokaryotes is 70S. In eukaryotes, the large subunit is 60S and the small subunit is 40S. Together, the rRNA is 80S. This difference creates drug targets for antibiotics. The antibiotics can target the rRNA of prokaryotes and destroy it without interfering with the eukaryotic rRNA since they're different types. Another difference in prokaryotes and eukaryotes is their start codons. I kind of alluded to this earlier when we were talking about FMET. So eukaryotes use methionine or MET, M-E-T, as their start codon, whereas prokaryotes use N-formyl methionine or FMET. Next, there are differences in the basic enzymes used in prokaryotic and eukaryotic transcription and translation. The first and main difference is in their types of polymerase. In transcription of prokaryotes, RNA polymerase is used instead of DNA polymerase, like in eukaryotes. RNA polymerase is quite different from DNA polymerase, besides the fact that it works with RNA instead of DNA. First off, it does not require helicase like DNA polymerase does. DNA polymerase would not be able to perform its job without helicase, whereas in RNA polymerase, that extra enzyme isn't needed. Secondly, RNA polymerase is a lot slower than DNA polymerase and needs uracil instead of thymine, like in DNA polymerase. Since this is the case, before RNA polymerase can do its job, all thymines need to be turned into uracils. Lastly, RNA polymerase lacks any proofreading function like in DNA polymerase. This leads to more errors in the process of prokaryotic transcription. This also means that there are no introns, RNA splicing, poly A tails, 5' methylated caps, and no RNA processing in prokaryotes. Lastly, since there is no proofreading, there is only one exon involved, which is vastly different from the process in eukaryotes. Next, there is this individual enzyme that is not seen in eukaryotic transcription or translation, and it's called E. coli hollow enzyme. E. coli hollow enzyme is the core enzyme for E. coli transcription. It is saturated with sigma factor 70, and it initiates RNA synthesis from sigma 70 specific. It initiates RNA polymerase from RNA synthesis, from sigma 70 specific prokaryotic promoters. Next, we're going to talk about why the location of transcription results in key differences in the process of transcription and translation. So in prokaryotes, transcription and translation both occur in the nucleoid and in the nucleus of both transcription is located in the nucleoid of prokaryotes and in the nucleus of mitochondria. The translation is in the nucleus of prokaryotes as well, but however, in eukaryotes, the translation is in the cytoplasm. This location difference is huge as to why transcription and translation is different between prokaryotes and eukaryotes. For starters, the process of transcription and translation is much faster in prokaryotes than in eukaryotes. This is because both processes are located in the same area and don't have to worry about going to different locations to perform the other process. This is also because prokaryotic transcription has no proofreading steps, but many more mistakes and mutations that don't occur within eukaryotes. This was mentioned earlier on the last slide. Also, in prokaryotes, translation does not start when transcription ends. Translation can begin on the same strand that is being transcribed after a part of it has finished, which was that coupling of transcription and translation we discussed earlier. So the strand goes through transcription and translation almost simultaneously. As soon as the RNA polymerase is done with part of the strand, translation can begin immediately. In eukaryotes, this isn't the case. Translation doesn't begin until after transcription and all the proofreading mechanisms are completely finished. Lastly, prokaryotic transcription incorporates polycentronic messenger RNA, whereas eukaryotic transcription does not. Polycentronic mRNA has the ability to transcribe several genes encoded in a single transcript. Transcription in prokaryotes does not have to transcribe to just one gene at a time, which significantly increases the speed of transcription. Now that we have fully discussed the processes of transcription and translation and discussed the key differences between transcription and translation in prokaryotes and eukaryotes, it's finally time to practice translation in real time. So I'm going to be demonstrating how to translate a real prokaryotic gene. The gene that I will be using is superoxide dismutase, which is seen in the H. halobium Mn containing superoxide dismutase gene. So it is important that you pay close attention to the steps because afterwards you'll be doing this on your own with a made-up gene. So to first begin the process of translation, you first need to discover where the open reading frame is because you will not be transcribing the whole gene. So as shown right here in this image, I have given you a whole gene, 5' to 3' everything. However, we will not be translating all of this. We're only going to be translating the part highlighted in that brown color because in the brown is that open reading frame. But also there are a couple other sequences highlighted too that are important to know. That +1 gene that I was talking about, which signals the start of transcription, is this blue right here, this blue G. That -10 region and -35 region is seen right here with the pink and the green. This is where that TATA box is located, where that sigma factor 70 will come and bind. Next, also, I didn't really mention this earlier, but there's also something called the exclamation point region, which is this purple thing here. This is just basically signaling the end of the open reading frame. That's all that is. So that's right here in purple. So now that we have located our open reading frame right here in brown, it's time to actually begin the translation process. So first, we're going to isolate the open reading frame, like I did here. It's important that you know what an open reading frame and to isolate it so that you do not end up transcribing the entire gene or translating the entire gene because you do not need to, and that's actually quite wrong. So now as you see here, all that we have is that brown highlighted region or open frame region. So now it's time to begin. And as you can see at this front, the open reading frame will always begin with an ATG in the DNA right here. However, as I mentioned before, and some of you might remember, we cannot work with ATG. RNA polymerase will not be able to read ATG. So now we need to turn all the thymines into uracils before we can begin any translation, because without that, RNA polymerase cannot read it and we cannot begin the job. So it is now time to do that. So translating to RNA. So what we're going to need to do, like mentioned before, is we're going to need to turn all our T's to U's. So our first right here is we see an A, but then we see a G or a T, so that needs to turn to U and G. And look at that, we have our start codon. So that's how you know you're on the right track. Next, we can just continue writing all of them down like so until we get to another T. There we go, right here. So we're going to keep going like this until all the T's are turned to U's. However, that's going to take a bit long. I think you guys get the idea, so we're going to go ahead and speed this up. So on this next slide, all of our uracils or our thymines have turned to uracils. As you can see here, we have only U's in our sequences, no more T's, which is good. So now it's time to translate the RNA to our amino acids. So what I think is easiest, and what a lot of students find easiest, is once you translate the thymines to the uracils, and when you get that RNA sequences, it's easiest if you divide all of it into groups of 3, because as we'll see later on, all of the amino acid sequences are in little triplets. So if you go ahead and you divide the sequence into triplet sequences, it will be easier later on to match the correct amino acid sequence and not get confused. So I'm going to demonstrate that now. Also, I forgot to mention, but in the regular gene, original gene, it was in that 5' to 3' position. Same with the RNA. We are still in that 5' to 3' position. And that's an important factor to point out, because we will not end in the 5' to 3' position. Amino acid sequence does not contain a 5' to 3' position. That is no longer a thing. So now it's time to divide all of them into groups of 3. So our first group of 3 triplet is going to be always our start codon, AUG. And now basically you just go through that sequence like so, and you just divide them into groups of 3 so that later on it'll be easier to read. And if you want to do what I do and you want to underline as you go so that you know that you correctly are translating, or not translating, but correctly putting all groups of 3, that might be useful because it's easy to make mistakes. It's a very tedious process. So now that you get the idea, I'm going to go ahead and fast forward because you don't need to watch me write the 3 pairs of all of this. So now we have everything divided into our triplets like so, still in that 5' to 3' position. It's now time to finally move on to matching the correct amino acid sequences to our triplet pairs. So this will be the final step of translation. And it's also maybe the most important step because this will be your final answer. So it is important that you correctly match each triplet to each correct amino acid. And you are always going to start with AUG, and you're always going to end with our stop codon, which as you see here is either UAA, UAG, or UGA. As soon as you reach one of those triplet pairs, even if you're in the middle of your sequence, you stop. You do not continue going on. A lot of students make that mistake. They see the stop codon, they write down the word stop. However, they realize that they're not technically done translating the gene. They still have triplet pairs left over and they continue going. However, that is wrong. Once you see that stop codon, you stop, because in the process of translation on an actual gene, which we kind of discussed earlier, the process does not see the stop codon and keep going. It stops. The cell stops. It does not continue translating anymore. So now I'm going to demonstrate how you just simply use this chart right here to find match the triplet pairs to the amino acids. So our first one is AUG. So you're going to go over to your A, and you're going to try to find, your A, and you'll try to find where AUG is, which is right here. Start. Perfect. You can also see right here that it says methionine, or down below, FMET. In prokaryotes, it's FMET. So you want to make sure that you write FMET for the first amino acid sequence. Now, however, if you experience AUG later on in this sequence, which I think it's here somewhere, there's another AUG in this sequence, you are going to write MET, not FMET. FMET is only for the start. MET is for any other time you experience AUG within your sequence. So next one is AGC. So you're going to go over to your A again. You're going to go to your G, and there you go, AGC, which is SER, and you're going to write that there. Next, you're going to find your C, AG. So your C's are right here. And next, you try to find the one with the A, and right there, CAG, and you write that down. And you're going to continue doing that with all of them as you go along. And some of them have the same triplet pair for the amino acid sequence right here. Like serine, SER has 4 triplets that it could be to become serine. So just like arginine, 4 of them. Not all triplet pairs code for a different amino acid sequence. And also another really cool thing about the amino acid sequences is it's universal. All organisms share this same chart right here, which I think is really, really cool. We share it with bacteria, we share it with other animals, we share it with everything. It's very universal. So I think that is great. So now I think you get the idea of matching the triplet pairs up to the correct amino acid sequence. I will show you what it looks like when you finish matching all of them. So this right here is our final sequence. It's kind of a lot. This was it was a larger gene. The gene that you'll be translating yourself at the end of your worksheet is not as long for practice, but this is a real gene. It's reality. The one that you'll be doing is made up. So right here, like I said, it's that F met for prokaryotes, not met. That is wrong. If you write down met for a prokaryotic translation, it's immediately wrong. Prokaryotes use F met, and I think that's really important that you understand. But like I think I can find a methionine here somewhere. But if you ever experience right here, AUG, you're just going to write down just this met because it's no longer informal methionine. It's just methionine if you experience it in the middle of the sequence. And right here, this was our stop codon, UGA. I did not continue any more of the sequence past this because once we reach stop, we stop. So I think that really sums up our discussion today on the processes of transcription and translation. Thank you for listening, and I hope this was helpful in your understanding. It is now time to go back, if you haven't already, and complete the associated worksheet.